A new chapter, Animal Kingdom. When you look around, you will observe different animals with different structures and forms. As over a million species of animals have been described till now, the need for classification become all the more important. The classification also helps in assigning a systematic position to newly described species. Basis of classification. In spite of differences in structure and form of different animals, these are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, patterns of digestive, circulatory, or reproductive systems. These features are used as the basis of animal classification, and some of them are discussed here. Levels of organization. All members of Animalia are multicellular. All of them do not exhibit the same pattern of organization of cells. For example, in sponges, the cells are arranged as loose cell aggregates. They exhibit cellular level of organization. Some division of labor activities occur among the cells. In coelenterate, the arrangement of cells is more complex. Here the cells perform the same functions arranged into tissues, hence is called tissue level of organization. It still higher level of organization, organ level, is exhibited by members of Platyhelminthes and other higher phyla where tissues are grouped together to form organs, each specialized for a particular function. Each is specialized for a particular function in animals like annelids, arthropods, and molluscans, and echinoderms and chordates. Organs have associated to form functional systems, each system concerned with a specific physiological function. This pattern is called organ system level of organization. Organ systems in different groups of animals exhibit various patterns of complexities. For example, the digestive system in Platyhelminthes have a only single opening to the outside of the body that serves as a both mouth and anus, hence is called incomplete. A complete digestive system has two openings, mouth and anus. Similarly, the circulatory systems may be two types. Open type, in which the blood is pumped out of the heart and the cells and tissues are directly bathed in it, and second, closed type, in which the blood is circulated through a series of vessels of varying diameters, arteries, veins, and capillaries. Symmetry. Animals can be categorized on the basis of their symmetry. Sponges mostly asymmetrical. Any plane that passes through the center does not divide into two equal halves. When any plane passing through the center of axis of the body dividing the organism into two identical halves, it is called radial symmetry. Coelenterates, ctenophores, and echinoderms have this kind of body plan. Animals like annelids and arthropods, where the body can be divided into identical left and right halves in only one plane, exhibit bilateral symmetry. Organism into two equal halves, and this is known as radial symmetry, and identified as a left and right equal ones, this is known as bilateral symmetry. Diploblastic and triploblastic organisms. Animals in which cells are arranged into the embryonic layers, an external ectoderm and internal endoderm, are called diploblastic animals. Example, coelenterates. An undifferentiated layer, mesoglia, is present in between the ectoderms and the endoderm. And we can see the diagram 4.2. The outer one is known as the ectoderm and the inner one is known as the endoderm. The middle one is known as mesoglia. And here we can see that in triploblastic we know it as mesoderm. The animals in which developing embryo has a third germinal layer, mesoderm, in between ectoderm and endoderm, are called triploblastic animals. Example, Platyhelminthes to chordates. Coelom. Presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity which is lined by the mesoderm is called coelom. Animals possessing coeloms are called coelomates. Example, annelids, molluscans, arthropods, echinoderms, and hemichordates and chordates. In some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouches in between the ecto and endoderm. Such body cavity is called spudocilum. The animals possessing them are called spudocilomates. Example, Aschelominthes. The animals in which the body cavity is absent is called acylomates. Example, Platyhelminthes. The white colored one in the figure A is known as the coelom. And while coming to the B, there is pseudocoelom, the scattered pouches. And in the figure C, there is no coelom, known as acylomate. Segmentation. In some animals, the body is externally and internally divided into segments with a serial repetition of at least some organs. For example, in earthworm, the body shown this pattern is called metameric segmentation, and the phenomenon is called as metamerism. Notochord. Notochord is mesodermally derived rod-like structure formed on the dorsal side during the embryonic development in some animals. Animals with notochords are called chordates. The animals which do not form this structure are called non-chordates. From porifera to echinodermate are non-chordates. Classification of animals. The broad classification of animals are based on the fundamental features as mentioned in the preceding sections. In the figure 4.4.